Great. Well, it's a real pleasure to be here with you in Boston. As Dominic says, I've been slowly becoming more involved with PURA syndrome over the years. My background is in clinical genetics as a clinician in Southampton in the UK. And I'm just aware that there's not a great number of us clinicians around. And so I'm going to put myself out there and say, if you have any questions about clinical genetics, diagnostic testing, things like that, I'm very happy to I'm here today and tomorrow, so please do ask me. But I'm here really more with my research hat on, and I've been given this title to talk about, can RNA help us understand Pura syndrome? And as I was putting these slides together, I realized this was really hard. But I'm going to have a go, and do feel free to ask me things. No, it's all right. Okay, that's all right. So this is what I want to try and tell you in the next little while. What is RNA? Okay, you've heard a bit about it, but I'm going to show you some more pictures. And what does it actually do? You've heard a bit about that, but there's more to it. And why on earth should we study RNA? You know, why bother? And actually, what has it all got to do with Pura syndrome? So I'm going to try and talk about that. And then a bit about how do we do it? What do we hope it might tell us? And then just at the end, I'll say something very briefly about treatments that are about targeting RNA, but just one slide about that. So what is RNA? Well, we've heard a bit about it today already. And RNA really is like DNA, but it's different. And so if you are a person, you may have Pura syndrome, for example. And of course, we know already that your body is made up of these cells. And hopefully this is by now familiar to you, that in your cells you have, in the nucleus, the center, you have the instructions, which is DNA wrapped up in chromosomes. And this is really like your library. So if I'm going to work with this analogy here, you think of your DNA in the nucleus as the library of instructions of how to make actually a human being. And that's your library copy. But RNA is actually a working copy of your DNA. So actually, your DNA in the nucleus, it's not a lending library. You can't take the DNA out and do stuff with it. You've got to make a copy, okay? And that's really what RNA is. So you start with your DNA. Here's the familiar double helix. You'll see it's got different colors there. Those are representing those different letters, and it pairs up very nicely, and that's how you can copy it and have a near perfect match in every cell of your body. But when you want to do something with it, when you want to read those instructions, your cells have to take a copy. They have to make notes. And for one gene, say that's the Pura gene there, it makes one copy in this molecule called RNA. And you'll see it looks very similar to DNA, but it's one strand, okay? It's just a single strand. And it's very slightly chemically different. And then that gets trafficked out into the rest of the cell to then be read by this process called translation, where the cell then uses stuff that's lying around, amino acids, to build the protein that fits the instruction. And that's essentially what's going on. I'm trying to figure out analogies here. So you start with your library, okay, and you make some notes, okay, you can transcribe it, it's transcription, and in fact then you need to condense it into something. So for example, you might get a recipe for something, okay, and I know it's just before lunch, so I've put this in. You might have a recipe, okay, but that's not the finished product, okay. Now you have to, as a cell, you need to pick all the ingredients out and make it into the real thing, okay, and then you've got something. And so this is apparently what French onion soup is. So that is sort of what's going on in a nutshell for each of your 20,000 or so different genes. They're doing different things with different recipes at different times in different cells. So it's quite complicated, but that's in a nutshell what's going on. And if you look at it in the cell, then you'll see there's the big purple nucleus. And what happens is the copying of the notes and things goes on in here. And then you send out the notes, and they get processed a bit, and then in the rest of the cell, in the cytoplasm, these other little machines find the amino acids, depending on the sequence, and they put it together into a chain. And if you blow this up a bit, you'll see it's actually made of these little machines here, which are basically enzymes, and they read off the DNA from one strand, they send it out, and then other little machines, these ribosomes, they come along and put it together, and you see this little chain of amino acids growing and growing and growing as the thing goes along the strand, and eventually you get your protein. which goes off and does its thing. For example, Pura, or Pura alpha, the protein. And just to show you a picture that back in the 1960s and 70s, they looked at this to show you that it really does happen. And here's a very electron micrograph, very, very high-powered image. And you'll see there's a very thin strand here, which is one strand of DNA. And then as the transcription goes along, you get these little growing chains of RNA being made. And this is a slightly artificial setting, so actually the ribosomes are also attaching to the RNA and generating protein. You get these, you see this happens and makes a little sort of Christmas tree structure. So this is a sort of idea to give you the sense of how this is going on. So what does RNA do? Well, the main thing that we usually think about is how RNA is read in the way I've told you to make proteins. And this is a particular sort of RNA called messenger RNA, okay? But that is not everything. There are other types of RNA out there that do different things. I'll try to very briefly and simplistically summarize what those things are. So here we've got translation into proteins, the messenger, that's what we usually talk about. But also RNA is important for some of those little machines that are helping you in the cellular machinery and helping to make the proteins as well. Okay, that's all I'll say about that. There are other little RNAs out there and long ones that actually their role is not to be made into protein, but to actually recognize other RNAs and regulate them. So stop them from doing things, get rid of them, and they basically just interact with our other RNAs and they control how the genes are made and how they're expressed, how they do their jobs. And this is an important one here, because actually there's still a lot about RNA we don't know at all. And so this is really a growing field, and it makes studying it quite difficult. And so why should we bother to study RNA? I thought this might be helpful to think about. So I've just told you that every cell in your body really has this library copy of your DNA, and it's basically the same in every cell. But all of your cells are different, okay? Why is it then that all of your cells in each organ looks different? You're not just a big blob of things with the same information. And that's because, actually, imagine, go back to the library analogy. So one cell, or in one case, you might be interested in a certain topic, okay? But in another setting, someone else might be interested in something else, okay? So in the first instance, you're going to go and look at these genes and work out their recipes and what they're going to do. But in another tissue, another cell type, you're going to be interested in other copies from the library. And other people, actually, they've gone to the library to find something for their child to read. Other people have just gone there to work. And people go there for a course. So, in fact, there are many different ways in which you can use the information stored in your DNA, in your library. Okay? And that's basically what's happening in your different cells, in your different organs. Different genes are active and making RNA in these different tissues. And that's why, part of why, everything can have the same instructions underneath, but you end up with lots of different organs and tissues. So what's this got to do with Pura syndrome? Well, if you have a medical condition of some kind, then that's very likely to influence how your genes are being used. Okay, for example, in your brain, we suspect there's going to be some change in how people with Pura syndrome, how their genes are being used, because the Pura and Pura alpha protein is so important that it's bound to be regulating lots of other genes. And we'd like to try and understand that. And in the rest of the body as well, these other tissues probably, and by having the condition, and also by having things like epilepsy and other problems, those are going to influence how the genes of the genome are used. And you can see that by looking at RNA. So it may show up. If you looked at the RNA, you can say, well, I know this is a very important gene, for example, in the brain. Is it being made as much as it should? Or is it actually absent? And why is that happening? And is it being processed correctly? Because it's quite a complex process to make the RNA and to process it. And we hope that by studying the RNA in model systems and also hopefully in patient samples, that we can try to find a signature that is different in PURA from in healthy people. And this is what you might call a biomarker. So you might have come across this term biomarker. It's basically something you can measure that is a proxy for a condition, some sort of readout. And this is across medicine this happens. So it doesn't even have to be something like a small molecule. It could be your blood pressure or something might be a biomarker for your cardiovascular health. And things you measure in your serum can be a readout for your kidney function, for example, like that. So some biomarker, and we feel this would be really nice if we could do this in PURA because first it might help us to understand the disease, and also, hopefully, when we come to a time when we can have some sort of treatment effect, you could also potentially measure this and say, oh, this is getting better, therefore this is working. So there's many nice reasons to try and work out what is a biomarker, and RNA might be one option. So I just put in some pictures of where I work, because how come I'm here? Why am I doing this? Well, this is Southampton, and this is the hospital in Southampton. This is the maternity hospital where the genetics department is. And I also work in the University of Southampton, which is co-located on the site. And I work with Professor Diana Borelli. Diana's particular interest is in RNA, and particularly in something called RNA splicing. And that's what we've been trying to look at. So I've shown you a bit about how RNA gets used in the cell, but it does need some processing before it can be used to make a protein. And in fact, the notes that you copy out, they need to be stuck together and condensed into just the important bits. And so this is a cartoon of sort of what this looks like. So if you have a gene that is DNA, there are some bits that are what we call the coding sequence. These are called exons, little boxes here. And then there are bits in between called introns, okay, which do not directly code for proteins. So these are the bits that are particularly important for coding the amino acid sequence. And when the RNA is made at first, it contains everything, okay? But we don't want all of that. We actually want to get rid of these little bits in the middle, and this is a process called splicing, where you cut out the introns and you stick together the exons like this, and then you have a finished RNA product, a messenger RNA, and that is what gets made and translated into protein. So this is a very complicated process, and I just put this really complicated slide to say that splicing is complicated. There are lots of factors that influence it. Other proteins come and bind to the RNA. Depends on the DNA and how that's wrapped up. Many, many factors, many of which we still don't really know. But one thing we wonder is, could actually Pura and Pura Alpha be part of this process? Because we know that some of the proteins that are involved in this process do associate with the Pura Alpha protein. And therefore we wonder, if Pura Alpha is not there as it should be, then there's some imbalance in how the splicing is working. And could that be part of the reason why things go wrong? Okay, so how do we actually study this? What do we do? It's kind of complicated, but I'll just talk about two particular things. So RNA is, as I've said, very similar to DNA, and so you can do some things that are analogous to looking at DNA. You can look at specific RNA molecules and to assess, are they as long as they should be, and are they there as much as they ought to be, the abundance, okay? And there are simple sort of molecular biology techniques that have been around for a long, long time, whereby you can see, is the RNA the right size? This is a gel electrophoresis, and here's some healthy bits of RNA, and here's a bit of RNA where something else has happened, and it's become shorter. And that's an indication that there's something not right about how it's being spliced together. In terms of quantifying it, there's techniques that actually try to measure the abundance of the RNA to say, is it being generated, is it being copied from the DNA as much as it ought to be? That's what I'm going to say about that. And then, more recently, in recent years, there's been a real revolution in how you can sequence things. And a lot of your families have, I suppose, come to a diagnosis through these techniques applied to DNA in the form of exomes and genomes. But you can apply this to RNA as well, because RNA is just a copy of DNA sequence. You can also sequence it and try to find abnormalities that way. And there's a whole host of new machines and technologies that that are out there to help us look at this on a research basis. Seeing as Lena put up a complicated slide about the output, I put one in too, that this is the sort of thing you get out. And don't worry about this, but here is the Pura gene here, and this is in a mouse, so it looks a bit different in a human. But that's where we're looking at. Here's the sort of chromosome up here. This is where we're focusing on the chromosome. And basically, these are little bits of RNA that have been sequenced and then mapped back to the reference genome. And what you can do is count up how many times has this bit of RNA been seen. It's red and red and red and red and red. So is it there as often as it should be? And you'll see here's an intron in the mouse anyway. And you'll see there's little reads that span over here, that's cut in the middle, and they continue here. And you can count, is that happening as it should be, or is it abnormal? This is just a very brief example of what you get out. And you've got machines and computers that help you. So what do we hope that actually by looking at RNA we're going to find out? Well, as I said, we want to know, is there some sort of signature in Pura syndrome that can help us understand what's going on? In fact, does it give us any clues of how best we could try to treat people with Pura syndrome? And in particular, what might be really great, although we have no idea if this will happen, can we find a particular RNA abnormality by looking at the splicing or something else that perhaps we could try to fix and try to manipulate? And could that be a drug target that might have some beneficial effect? So what are we doing in Southampton about this? I just put here, we are also working at the Pura Registry, but Rebecca's going to talk about that, so I'm not going to talk about that. We're also been collaborating with Jennifer Gordon's lab to analyze these Pura mice brains and try to work out in the mice, is there some sort of abnormality that might be applicable to humans? That's really complicated, and that's ongoing, but we're working on that. And we're also looking at some cell biology to try and model the Pura gene in some cells to understand what it's doing in terms of the RNA. And don't worry about this, but I put this in here because it's interesting. So this is, if you look up Pura on a genome browser, you can find out interesting things about it, and if you know what it means. And here again is the Pura gene in humans, and it's an unusual gene because actually it's very short, and it's actually, you'll see, it's just one block. So in humans, it doesn't actually get spliced together. It's just a very small thing, but it has this very, very long tail here, which is not something that gets made into protein. This is the bit that gets made into protein. But this long, long tail is actually probably quite important. And we know that because if you overlay the Pura gene in many other species, down here to frogs and things, they also have exactly the same sequence in this bit that doesn't get made into protein. And we think this must be really important because, as Dirk showed us, there's hundreds of millions of years of evolution in between, and the sequences are almost identical. So there's something we need to find out about what this is doing at the RNA level, and we're hoping to do some work on that. So this is tricky and it's challenging, and I'll just say why it's quite difficult. Because RNA is dynamic, so your DNA library is always the same. Every cell, doesn't matter, it's always going to be the same. But RNA is changing. Okay, it's really like the weather. Okay, and we are trying to really understand what is happening, what's the climate like in these cells, but we're only able to measure at single points. What is the weather like, and therefore what is the climate? It's quite difficult. In fact, for meteorology, you have the most powerful computers in the world trying to model what's going to happen in a few days' time. It's very difficult. So it's actually really challenging to study RNA like this. But we'll have a go. And a couple more analogies to leave you with about what this is like. So DNA is a bit like taking a picture or painting a still life, okay? Everything is always in place. If you come back tomorrow, it'll basically be the same. That's sort of what it's like. But looking at RNA is more like looking at a snapshot in time. And if you came back tomorrow, this will not be here. And so this makes it really difficult to get a good overall picture, unless you have thousands of samples, lots and lots of samples to say what's going on. And another similar thing, okay, you could say the genome, looking at the DNA, is a bit like looking at a map. Here's a picture on Google Maps. I believe this is in San Francisco, the stadium, and you could even have some Google Maps with photos. But then if you actually send a satellite out to go and look at this picture, you might say, well, is there something different about this? Well, there's a couple of little things that are different from the map, but it's basically the same. But if you were to look at the RNA, it's a bit more like saying, is this thing being used as we would expect? And here, the stadium, which should be for football, is actually being used by WrestleMania. So it's a bit more like, it's not always what you expect, and this is trying to say that RNA can be used different. It's different, and it's not always what you expect. So the final thing I wanted to talk about briefly was, we hope that we might find something that is of real relevance in terms of something that could be worked into a therapy. We have no idea if that's really going to happen for Pura or not by looking at the RNA, but we'll have a look and we'll try and see if we can find that. And some of you might be aware of some new treatments that are coming along for certain genetic disorders that target RNA. And there are two types of therapy at the moment that are sort of manipulating RNA. One is where if your RNA is doing something toxic, and we don't know at all if that's what's happening in Pura, but then you can actually make these little antisense drugs that recognize specific RNAs and knock them down, degrade them. And that's being used in Huntington's disease at a clinical trial phase, and also, rather excitingly, for motor neuron disease, for ALS. And that's one approach if your RNA is toxic. And another a way that has a little bit more advanced, at least for a couple of conditions, is by correcting splicing. And you may have come across spinal muscular atrophy and a drug called Spinraza or nusinersen, and that is targeting a very specific and very well characterized splicing abnormality and correcting it. And that has had really dramatic results and really revolutionized the care of these patients. Duchenne muscular dystrophy is another disease that sometimes is related to splicing, and there is at least one drug that has it has been approved, and it can correct the splicing and have some effect in the disease, but it's not as dramatic as in spinal muscular atrophy, and that's because the diseases are different and the targets are different, and so it's very individualized treatments. And so whether or not this will ever be something applicable to Pura, we don't know. But if we can find something, then that would be great. Just to illustrate what's happening is you have your RNA target, you have an antisense drug that recognizes exactly the place that you want to target, and then depending on what it is, it can either degrade the RNA or manipulate the splicing in some way. And so that's really all I really wanted to say. I'm part of Diana Bray's group, and this is us at the moment. In terms of the RNA work, we've also been working with Marco Borelli, who is Diana's brother, who also is interested in splicing in Italy. And of course, Jennifer's been giving us mouse brains. And these are the people who've been funding some of the work. So that's more or less all I wanted to say. I'm very happy to take any questions.